Long time no see. Today I'm going to discuss how I interpreted blood chemistry and the blood cell count laboratory reports. This is video 1 in a 3-video series that I'll be attempting to discuss how I interpret blood chemistry and blood cell count reports. When presented with a sick dog or cat, a comprehensive history taking and physical examination is what any veterinarian does. If the history and clinical signs do not lead to a conclusive or tentative diagnosis, I'll seek authorization from the client to perform blood work. Blood analysis is comprised of complete blood cell count and blood chemistry. Blood analysis may offer a better picture of what is ailing the dog or cat or confirm a tentative diagnosis. Clients are always eager to know what their pet's blood results have revealed. In this series of videos, I'll attempt to discuss how I interpret blood results in a way that the client will be able to understand them. Blood chemistry and blood cell count are reported separately, but I will attempt to integrate them as much as I can for the purpose of easy understanding. This is how blood chemistry and blood cell count looks like. Right? For the blood cell count, it starts with the glucose, electrolyte, sodium, potassium, chlorine, carbon dioxide, sodium and potassium ratio, anion gap, serum osmolarity. It looks at the kidneys, urea, nitrogen, and creatinine. Then we put blood urea, nitrogen, and creatinine ratio. Total proteins reports the total proteins, albumin, globulin, and the ratio. Potassium, calcium, phosphorus, chlorine, phosphorus ratio. Bilirubin, total, direct and indirect. Alkaline phosphatase, the 3 liver enzymes, alanine aminotransferase, aspartate aminotransferase, and GGT, creatine kinase, cholesterol, triglycerides, amylase, and finally thyroid studies. On the blood cell count, it starts with leukocytes, red blood cells or erythrocytes, reports on hemoglobin, hematocrit, MCV, MCH, MCHC, segmented neutrophils, bad neutrophils, and the lymphocytes. And then the monocytes, eosinophils, and basophils. The white blood cells are reported in 2 ways, either as a percentage of the total white blood cells or in the thousands per microliter, right? All those are repeated again. And then at the end, you have the platelet count. Blood constitutes 7 to 9% of a dog's body weight. In cats, blood constitutes about 6% of the body weight. Blood is comprised of 4 components: the plasma, white blood cells, red blood cells, and platelets. Plasma is the liquid portion of the blood. Plasma transports lead and white blood cells, platelets, proteins, nutrients, and other secretions to all parts of the body. Plasma is involved in the regulation of the fluid balance in the body. Let's see where this one is next. This is a whole blood. If you centrifuge whole blood, you are going to get 3 layers. You get the plasma, which consists of 55% of the total blood. Then you have the buffer coat, which normally contains leukocytes and platelets, which forms about less than 1% of the total blood. The rest is red blood cells or erythrocytes. They form about 45% of the total blood. The red blood cells carry a protein compound called hemoglobin. Hemoglobin is responsible for the red color of the blood. The red cells transport oxygen to all tissues of the body and returns with carbon dioxide for exhalation from the lungs. The white blood cells or leukocytes defend the body against harmful agents like bacteria and viruses. The platelets are involved in blood coagulation or clotting when the veins, arteries are injured. When we draw blood from a dog or cat for complete blood count analysis, we place it in a test tube with an anticoagulant to prevent clotting. For blood chemistry and serology, we place the blood in a red test tubes without a gel separator. All measurements of blood components are reported as a range between a low normal and a high normal. The low normal and the high normal. Low normal, if you look at leukocytes here, the range is between 6,000 and 13,500, right? 6,000 is the low normal, 13.5 is the high normal. Look at the glucose here, measured in milligrams per deciliter. The range is 77 milligrams per deciliter on the lower edge and 155 milligrams on the high normal. Okay. The actual and the normal values are provided on the first column. Right, that's the first column here. It reports the normal values which the laboratory have found after measuring it. Measurements falling outside the normal range, are reported on the second column as high or low or critically low or critically high. If you look at the urea nitrogen here, it was critically high, 123.4 is not supposed to go beyond that 6 milligrams per deciliter. But this was almost 4 times high of the high normal. The measurement units are reported on the third column, right? This is the third column. Right? That's the third column, that's the measurement column. It shows with what units the parameters have been measured. The range is provided on the fourth column, which is this one here, right? Which is this one right here. Laboratories format and report their analysis results in nearly the same way. Blood chemistry results are reported first, followed by the white and red blood cell count. All laboratories will alert you of any changes like hemolysis of the blood specimen that may affect the results. In this blood work, the chemistry report starts with the glucose and ends with thyroid studies. Glucose ends up with thyroid studies. The blood cell count starts with the white blood cells right there and ends up with the platelets down here. That's the end of this video. If you have liked what you have heard or watched, you can subscribe, share it, comment, or ask a question below. Thank you for watching. In the second video, I shall try and discuss or analyze the white blood cells and try to integrate the blood chemistry results with the blood cell count report. Thank you for watching.